Thanks, Dr. Merritt. Next up is Dr. Sal Coniglio, who actually works now closely with Dr. Merritt on the next stage of this cancer research. So we'll hear from him next.
Thanks. So I'm Dr. Coniglio. I've met some of you already. If you have a hard time remembering that, it's rabbit in Italian, so you won't forget that, okay? So my background is I have a PhD in cellular and molecular biology from Albert Einstein College of Medicine, and I did my postdoc there as well, and I've been working on cancer cell invasion and metastasis for the last 15 years or so. And what my RFI stream does is we try to mimic how a tumor behaves in a dish as closely as we can to the situation in a patient. Okay, so when Dr. Merritt talked to you about screening new drugs, we could save a lot of energy and time and effort if we could find drugs that we can predict would work better in a patient if we could do that in a dish. Okay, because when you're looking for new drugs, you're oftentimes looking at thousands upon thousands of different molecules. So you have to have a way of kind of culling which ones are going to work and which ones aren't. And the better screening methods you have, the more likely you are to be successful. So the long and sad history of drugs that work really well in a dish but don't work so well in patients. And what we're trying to do is model that better. And what my RFI stream is doing is we're making something called tumor spheroids. And these are cancer cells that we grow in standard culture conditions. And we make these three-dimensional structures that look and resemble, for the most part, glioblastoma tumors as closely as we can get them to grow. Another thing we do that's very exciting is that we see how these cancer cells interact with normal cells of the body. So it's known that very aggressive cancers like glioblastoma, like metastatic breast cancer, they are filled with quote-unquote normal cells from your body, okay? And for the longest time, people didn't know what those cells were doing there. And it turns out that those cells are playing a very, very important role in allowing the tumor to progress and metastasize and evade the immune system. And we're generating these spheroids in my RFI stream that they have. You guys who just did my session, we're coloring those cells individually, and we're tracking what they're doing in these spheroid cultures. And the hope is that we can add combinations of drugs that can block different targets and can work in a dish, and then eventually take that into animal models and maybe someday the clinic. So, okay. See you soon.